Good evening. So today what we're going to do is we're going to go over the structure of the DNA. We're going to learn about what does the DNA do and how do we get that double helix structure of the DNA. The first thing that we're going to do, we're going to look at the function of DNA. What does it do for us? These are the learning objectives. So we're going to learn about the role of the DNA as a storage and a copying mechanism of information. We were going to look at the history of the DNA. We're going to look at the events that led to us understanding what is the structure of DNA made from. We're going to look at the structure of the DNA, what is it made up from. We're going to look at the different bases that make up a DNA, and we're going to look at how the sequences are paired to build a DNA molecule. Now, the first thing that you need to know is the role of DNA. The DNA has 3 jobs, I must say, in our body. The first job is to store information, which is the genetic information, to copy that genetic information and transform the genetic information from one cell to another. Remember, we constantly go through the process called mitosis, where we basically grow and repair our cells. So the new cell that we have forming needs to have genetic information as well. So that genetic information is passed on in the form of DNA. So the first thing that you need to know, the DNA is the storage of all the genetic information that is needed to build an organism. So your hair color, your eye color, your height, your weight, even your personality, the genetic information that codes for these things is stored in the DNA. So the first thing is to store, okay, to make sure that it's safe, that we have the master copy, okay, the big book that has all the information that we need. The second thing is, the second job of the DNA is to transmit information. Like I told you, when a cell divides, the new cells must have the same genetic information that the parent cell has. So in cell division, or in the process of mitosis, or before mitosis happens, remember, if you remember interphase is the stage where the DNA replicates or doubles. This is to make sure that the new cell that we're going to form has the same information. The next thing that you need to know is, what is the DNA made from? Look, you find there's a lot of molecules, biomolecules that we have in the human body. Some of them are proteins, we have carbohydrates, we have lipids, and we have nucleic acid. Now, we know carbohydrates is a source of energy. We know that we use lipids to store energy and to give us insulation or to keep us warm. We know that we use proteins to build muscles, to build hair, etc. But what we don't know is that actually we use something called nucleic acid to build the DNA molecule, okay? Each nucleic acid is made up from small units known as nucleotides. I'll show you in a bit. Right, so as you can see here, all right, this is the structure of DNA. You have the phosphate group, and in the middle we have a sugar called known as deoxyribose, and then we have a nitrogen base, okay? Now, one phosphate group, one sugar, and one base, that's a nucleotide. Here, we have 4 types of bases in DNA. We have adenine, guanine, cytosine, and thymine. These are the 4 types of bases that are found in the DNA. Each base is attached to a sugar and phosphate backbone. Now, the sugar and the phosphate and the base are attached to each other through a bond known as covalent bonding. Now, if you remember in chemistry, covalent bonding happens between two compounds or two molecules which are non-atoms. Now, what you need to know about the nitrogenous bases, okay? Now, what you need to know that adenine and thymine can be attracted to each other. Guanine and cytosine can be attracted to each other. That's how we have a double strand of DNA. Now, also again, let me remind you, the nucleotides in the DNA, the sugar and the phosphate groups are joined together by covalent bonding, not hydrogen bonding. We'll get to the hydrogen bonding later. What you need to know is that you can join these nucleotides in different orders. So you can have an adenine next to guanine, next to cytosine, next to thymine, or you can have thymine next to cytosine, then guanine, then adenine, and so forth. So that's the structure so far. Now, if you know from previous subjects that you studied about the DNA, you know that the DNA consists from a double helix, two strands of nucleic acid attracted to each other. Now, before I explain to you about the structure of the strands, let's have a look at a little bit of history. Now, the first person or the first lady who tried to figure out the structure of the DNA was a lady called Rosalind Franklin. She was a British biophysicist, which means she used physics to kind of look into biological concepts. Now, she was the first person to be able to be successfully identify the structure of the DNA, the double helix. What a lot of you do not know, that unfortunately Rosalind Franklin died from cancer, I believe, due to the fact that she used to work with a lot of x-rays, and now we know that radiation is actually a cause of mutation, which can lead to cancer. So unfortunately, what she was working with led to her death. Rosalind Franklin didn't win a Nobel Prize for her work, unfortunately, because by the time they recognized her efforts, she already sadly passed away. Now, so we figured out that the structure of the DNA looked like an X-shaped, okay? Obviously, the technology wasn't too advanced back then, but she did manage to figure out that we have some kind of X-shaped pattern for the DNA. The other two that really were able to successfully build a model of the DNA were Watson and Crick. And actually, these two guys were able to build a model of the DNA, the double helix model that we know now. Unfortunately at the same time, Watson and Crick were able to win a Nobel Prize for their work into DNA. Unfortunately, Rosalind Franklin wasn't so lucky. So this is what you need to know. They were able to build a model of the DNA, the double helix model that we know, and this was largely because of the work that Rosalind Franklin was able to do. So Rosalind Franklin was the first person to determine that the DNA had the structure of X. Watson and Crick used her work to build a model of the DNA. Another guy that we need to look at is someone called Chargaff. Okay, Erwin Chargaff discovered that the percentage of adenine and thymine are equal in any sample of the DNA. So what he meant that adenine, thymine, guanine, and cytosine, if you look at them in every DNA sample, you will find them in equal ratios, which led to believe that in the DNA you have adenine, thymine, guanine, and cytosine in equal amounts. Now, this is very important because in the DNA molecule, not all bases are attracted to each other. So, for example, adenine is attracted to thymine, and guanine is only attracted to cytosine. So, if we are building a DNA molecule, if you have an adenine in one strand, the opposite has to be thymine. If you have guanine on one side, this other side needs to have cytosines, and so forth. This is known as Chargaff's rule. Now, I'll explain to you now how that works. This is known as base pairing. So each two bases, they go together, okay? Adenine and guanine cannot go together. Thymine and cytosine cannot go together. If I show you this, if you look at the model here, or the graph here, or the picture here, you will see that guanine would only bond with cytosine. Adenine would only go and become attracted to thymine. Thymine would be only attracted to adenine. Cytosine would be only attracted to guanine. Okay? Now, the force of attraction between these bases is not a covalent bond, it's a hydrogen bond. The reason why we have hydrogen bond, because it's important that we are able to break the DNA strand, because we sometimes need to copy the DNA strands to make them replicate and so forth. So it's important that the attraction between the two strands is not a strong covalent bond, it has to be a hydrogen bond. If you go back a little bit, the model of the DNA that we know is called the double helix, which means you have, in each strand, you have a phosphate backbone, a sugar in the middle, and a base, okay? The two strands are joined together by something called the hydrogen bond. And this is what the cross-section of the double helix looks like. Now, again, remember, base pairing. Guanine pairs with cytosine, adenine pairs with thymine, and so forth. We have hydrogen bond forming between the two base pairs. Why do we need hydrogen bonding? Because we want to be able to break the two strands of the DNA when we want to replicate them. When we want to replicate DNA. This gives you an idea about the structure of the DNA. So what you need to do is you need to focus on the structure. You need to focus on the people that did work to understand the nature of the DNA and what function does the DNA do. So remember, we have three functions: copying and storing, two functions, copying and storage of information. We need to look at what Franklin did, Watson and Crick. We need to understand the basic structure of the DNA. We have a phosphate group, a sugar, and a nitrogen base, and how we use base pairing rule to build up a DNA molecule. I hope this helps, and thank you for watching.